still as a fairly young man, what do? You're always the Thursday night, dinner speaker at the conference. And he liked to get up and say, I've just thought about a problem that none of you have thought about yet, but I want you to hear what I'm scribbling. And these are actual scribbles from his lecture that are in his delightful environment. So he thought about, what's it like for a bacterium to be swimming around? How does that compare to, say, us as a swimmer? Well, the important number is the Reynolds number. The Reynolds number is the ratio of the inertial force, F equals ma, divided by the viscous force. For big things like us, the inertial force dominates, okay? And if we dive into the swimming pool, we coast for a long way. If we row a boat with oars that push the water that way, we go that way. But for the bacterium, which is operating at a Reynolds number of 10 to the minus 4, that doesn't work. The coasting time is less than a microsecond. The coasting distance is sub-angstrom. So the bacterium can only move by continuously providing force. As soon as the force stops, the motion stops. And the efficient way to do this, according to Purcell, is to have a non-reciprocal motor. So if you have a clamshell, that's not going to work. It's going to do this and that and this and that, because the motion is reversible low limits. But a corkscrew works really well. So if you've ever uncorked a bottle of wine, you know that the corkscrew is a very efficient means of taking this very viscous object, cork, and transducing this rotary motion into a vertical motion that can make it go. And that's what the bacteria do so well, with an efficiency on the order of 10% free energy. So anyway, it shouldn't have worked, but it did work, and that's because another mechanism was making it work. And we knew something was wrong pretty much right away, because if you look at this video in color, which the camera didn't capture, you could see that actually it wasn't the gold end that's moving forward, it's the platinum end that's moving forward. So it's the opposite of what you predict from the jet engine model of oxygen coming off the platinum. What happens, and it took us a couple of years to pick this out somewhat painstakingly with lots of different metal combinations, is there are actually two electrochemical half reactions that add up to the decomposition of hydrogen peroxide. The anode reaction dominates the platinum. The cathode reaction dominates the gold. And that means that we build up extra protons over here, and we have a deficiency of protons over here. That creates a local electric field around the particle. The particle has a surface charge, which at neutral pH is a negative surface charge. And so this then just basically does electrophoresis in the electric field that is created via the catalytic reaction. So it's a self-electrophoresis mechanism. Now, the interesting thing about that, in addition to making these things move, is that the solution here is different from the solution here. And so hydrogen peroxide is depleted both front and back, but also this solution is basic and this solution is acidic. And that leads to a lot of interesting motor-motor interactions that we can study. And I'll show you a few examples of that, because these ionic effects, these electrokinetic effects, can be felt a long way away. So here, this is sort of the next generation that's looking at some of these collective phenomena. So what we've got here, this is a photochemical reaction with irregularly shaped sulfur chloride particles right out of the bottle. When you shine light on them, they make oxygen and hydrochloric acid and silver metal on the surface of these particles. And the diffusion coefficient of the proton is faster than that of chloride. So locally, that creates an electric field. And all these electric fields interact. What does that do? Well, on surface of the glass where we're looking at these things, the individual motion of the particle, individual catalysis creates convection moles around each particle. And those mutual convection moles draw them together like schools of fish. But because they all are bearing the same surface charge, they don't really like each other. They get so close, and then they stop. This is a predator-prey interaction where we now have a different kind of particle that's not catalytically active. It's responding to the electrical field generated by the catalytic levels. You see these tracers kind of surround the silver halide particles like an immune reaction. Maybe I run that movie over again. It starts out with this falling apart. This is if we switch the light on and off, and this shows you how beautifully reversible this whole cascade is. The explosion comes from the different diffusion coefficients of the proton and chloride making electric field, repulsive electric field, and you turn it off and everything reverses. So this is how we were recruiting students to the group showing this movie. We flip the microscope light on. And this is my favorite. This is the sharks and minnows game, where now we have an alkaline particle and all these negatively charged tracer particles running away from it, because the slow dissolution of the alkaline salt creates this electric field that makes the sharks and minnows. So the chemical motors do a lot of interaction with this. Okay. Now there's another interesting thing about these microswimmers, which is that they're slow, but they're about the same. This is the first generation speed. They're faster now, but they're about the same as a bacteria that are the same size. And if you think about us, we're much bigger, and cheetah is not much bigger than us, but faster. But there's a big difference in velocity between these two kinds of runners or swimmers. But if we normalize the body length, they're all about the same. And why is that? There's some simple physics, okay, that, so physicists have this, what they call the spherical cow approximation, where you first approximate the cow as a sphere, and then you do all the physics. And so let's analyze the physics in this book. Did anyone read this book, Ray? Just to catch you up, this is a book about how bad micro-robots can be, particularly if they can swarm and self-assemble, develop high intelligence, and then if they say, if their preferred behavior after that is either to eat human beings or to get in their brain and control them and make them do awful things. I don't want to ruin the end of the book, but the chemist is a hero in this book because the chemist knows the thermite reaction and blows up the nest. But anyway, the question is, could we actually make a fiendish swarm of flying silicon-based micro-robots that could really chase you down and eat you? And the answer is no. And that has to do with the physics. So the power that we can generate catalytically depends a little bit on the dimensionality. So for something really small, like our micro wires, this is a surface reaction, so this is going to depend on the surface area, which sort of goes as R2. For something bigger like us, think of us as like a bag of mitochondria generating power. If we're spherical, our number of mitochondria kind of goes as the cube of our linear dimension. So the power generated is a size to some power that's somewhere between 2 and 3. The drag force that that you experience going through a viscous fluid for small objects goes roughly as R to the 1, and for big things about R to the 2, like a friction force. So power is force times velocity. And so the power dissipated is the drag force times the velocity. And at whatever your top speed is, that's going to be equal to the power you can generate. And so you can then derive from that that this characteristic velocity is this r to the n over r to the m, which comes out about r to the 1, independent of size. And so this scaling law works basically for everything from the bacterium up to the jet airplane, within an order of magnitude or so. And so if you're being chased by these fiendish flying silicon micro robots, don't panic, just walk away. You're 10 to the 5 times faster. They'll never catch you. Okay, so anyway, understanding that as well as we did, and understanding how these electric fields work, we and others were able to develop some interesting applications. For one thing, using these electrokinetic effects, one can attract particles to, for example, places like a crack in a bone. This is not done in vivo, this is just an in vitro experiment. But if there's a crack, this is dissolving more ions than on the smooth surface of the bone. And with the right charge type, you can attract particles that fill that crack and glue it shut. A more realistic application is preventing fouling of membranes that are used for desalination. So in pressure-driven desalination, reverse osmosis, you're basically pushing salt water through these hollow fiber membranes, and you have fresh water on the inside, greatly reduced salt concentration. But at the surface of the membrane, you have a higher salt concentration than you do farther away as part of this dynamic process. The sodium ions diffuse faster than the chloride ions. This creates a local electric field that drives negatively charged particles to the surface. So if there are clay particles in the brackish water, they'll be impelled with this electric field to foul the surface. And this is a surface fouling problem. If you put alkaline particles like magnesium carbonate on the surface, they create hydroxide ions that basically push these clay particles the other way. So you can see the particles here, and there's this dotted line that indicates this area has been cleared by the surface coating. We could also make kind of smart gel pumps that, with enzymes that can sense sugar, can turn on the pump and basically expel whatever is being held in the matrix. So you put insulin in the matrix, and putting this in glucose solution causes the insulin to be released. And then this is a really cool one. This is not, all these three of these were from Penn State. This is one from another group at University College London, where they actually use the chemotactic force. So all of these motors chemotax, that is, they preferentially swim into higher concentrations of their substrate because the free energy is lower. If you have a binding site that's doing catalysis, the free energy is lower at high concentration than at low concentration. And the gradient of that free energy is a force. So there's effectively an entropic force that drives catalytic motors into higher concentration gradients of the fuel. So this was used by this group at UCL to drive vesicles across the blood-brain barrier, powered by glucose oxidase, the same enzyme, because the brain has much higher concentration of glucose than the bloodstream, so these vesicles can carry chemicals across the blood-brain barrier. Again, just in vitro at this point. Okay, so these were some of the emerging applications of chemically powered motors. And so, fast forward another decade or so, and everybody, lots of people got into the game. Okay, and this is always nice because people have a lot of new ideas. So people made, for example, motors that were maybe 10 or 100 times larger than ours, where the Reynolds numbers were quite so low, and then they could actually make the catalytic rockets that are fairly fast. This was a really interesting one. To make a corkscrew motor, this has a magnetic ball at the end, and then a helix here, and by turning the magnetic field, these things can be directed through fluids. This can also happen with light and with catalysts that, in this case, are decomposing carbonic acid to CO2 using carboxy anhydrase. But also, this is a photochemically sensitive system that can find which way the light is coming and point the motor out. And then this is a new one from my colleague Mark Michigan, where it's a photochemically activated corolla, where there's a light that's flashing on these little patches of semiconductor. That's powering these legs that are platinum foil legs that electroabsorb ions and go basically through electroabsorption, change their shape. And you can't see it because it's too fast, but it's actually walking in a kind of way like that. So there are all these different designs. And my favorite, though, because I love accidents, my favorite is one we discovered by accident. And this was kind of, I don't know, 8 or 9 years into this motors project. And I was on sabbatical. I was on sabbatical in France. Why in France? Because my wife told me to go there. She said, find yourself a gig in Paris. And I said, okay. I was thinking of going to Rochester or Cleveland. So anyway, we were doing some experiments with a physics group, a soft matter physics group at ESPCI in Paris, and they were interested in kind of the difference between our kinds of motors and bacterial motors, and so they needed a fresh batch of our rod motors, but I had a very bad student making them at that point, namely me. Okay, and so I played them too fast, and instead of having nice cylinders, I ended up with some that were concave on one end. This is the growth tip. The growth tip is pointy, and the other end is concave. Okay, and what they were interested in doing was they wanted to. This is Mauricio Hoyos, our collaborator there. He wanted to know how these are different from flagellar bacteria when they approach a surface. Because when a flagellar bacterium bumps into a surface, it's turning, the helices are turning this way, the head is turning that way. And when it gets near a surface, that actually creates a torque in the fluid that causes the rod to now cruise along the surface. And this is thought to be important in the formation of biofilms, for example. And so if you wanted to see if our motors that had a different propulsion mechanism would do this as well or would do something else. But because ours are heavy, they're made of gold, they had to be lifted off the surface. And you can lift them off the surface using ultrasound to create a cavity that creates an ultrasonic standing wave halfway up in this little swimming pool with a cover. And this is typically something that with a 100 micron gap, something like that, at ultrasonic megahertz frequencies. So here's a picture of how that works. There's a node right there, and typically particles are levitated in that node, and then you can hold them there and watch what they do. And you can turn off the ultrasound and watch as they sink to the surface. But we never got around to the experiment he wanted to do because we saw something very unexpected, which was now, instead of moving at 5 microns per second, these things were zipping around at at 200 microns per second. And these are the fastest and most powerful nanometers we've ever seen. Okay. And it took us a while to figure out how they work. As with the original chemical motors, we got it wrong the first time, but we eventually, with the help of some of our mathematician friends, figured it out. I'll get to that in a second. But the other cool thing about this was the ultrasonic propulsion again gives us autonomous motion of these little rods, but it's not chemically propelled anymore. We don't have to worry about screening the electric field with salt or anything. So these will work in any kind of medium and we don't need fuel. So you can actually put these in living cells. This is a HeLa cell. It's an immortalized line of cancer cells. And you suspend these with some of these gold microswimmers. And because these are the size and shape of bacteria, the cell thinks that it's being invaded by bacteria, and it swallows them by phagocytosis. But because they're made of gold, it can't enzymatically destroy them, and they end up in the cytoplasm. And then you can turn on the ultrasound, and basically these things run around the cytoplasm. There you go. Okay. And they wiggle around, and there's some that are zipping around the cell. They don't actually, these are pretty rough, tough cells, they don't actually hurt the cell. We do this for an hour or so. The same fraction of cells are viable as if we didn't do this. And so it's just kind of a toy. It happened to make a lot of newspapers because they were inside living cells, and this invokes a lot of fear when people commenting on your article on the internet. And so that happens, but I guess just spell the name right. But this is actually now kind of interesting for in vitro diagnostics. This is a paper from one of the many papers from, I think the first one of these from Joe Wong's lab at UC San Diego. They had a really clever assay that uses this effect. So basically, what they do is they take these acoustically propelled rods and they cover the surface with single-strand DNA. And then the cell ingests these particles, and the DNA is chosen to match the sequence of some low copy number RNA inside the cell. If they hybridize, it pulls the DNA off the cell surface, and there's a fluorophore that's quenched by the gold, but if it gets pulled off, then it lights up. And so this shows a comparison of the fluorescence of two of these cells, one in which the motor is taken up, but the power doesn't get turned on. And this is one where you turn the power on, and it runs around and basically collects every copy of this low copy number RNA and creates a big fluorescence. And then they have a whole bunch of other cool assays they're doing this kind of thing. We were much more interested in kind of the fundamental physics of how these things work. And so my student, Suzanne Ahmed, made a whole bunch of different bimetallic combinations. And we had done this before to understand the mechanism of the chemical propulsion. But these were quite interesting in the context of acoustic propulsion. So what she found out was the dominant effect in terms of the direction of these, say, gold ruthenium or gold rhodium, etc. The dominant effect is the difference in density between the two metals. And the secondary effect is the shape. So previously we've just been making them using shape asymmetry, but now she had density asymmetry as well. And it took us a long time to figure this out. We did a bunch of very systematic experiments that included 3 undergraduates, and some graduate student, and 2 mathematicians who are collaborators. And the mechanism that we initially proposed for the actuation of these things was basically a rattling mechanism, where one end is actually a higher Reynolds number than the other. But what they showed, and they showed this by fitting the data over a large parameter space of frequency, etc., was that the actual motion is more of a swimming motion, more of a dolphin-like motion. And that's actually consistent with the Purcell hypothesis about viscosity propulsion. Now, okay, so what can you do with these things? Well, one of the dreams of Feynman, Professor Schlatter mentioned this Feynman lecture. This is another quote from Feynman. It would be very interesting in surgery if you could swallow the surgeon. Okay. He had another great quote that was about physics, a little bit more audience. Do you know what that one was? Physics is like sex. Sure, there's a practical outcome, but that's not what I do. Okay, but can we get a practical outcome here? So in tumors, what's different about tumors from the bloodstream, normal bloodstream? Tumors are low, they're deficient in oxygen, they tend to be acidic, and they also tend to have high pressure. And so fluid is typically flowing out of tumors and into the vasculature. And the question is, can we use this effect, that is, knowing which way flow is going, and use it to direct our motors? So this was a really clever thing that my student, Mitsch did. He thought about this problem, thought about if I have a chemically propelled micromotor, remember it's generating acid at one end and generating base at the other. If the surface here is negatively charged, like a piece of glass, that's going to tilt, on average, this wire such that the positive end is a little bit closer to the glass than the negative. Now let's think about this in a flow. In a flow, we have the no-slip boundary condition of the surface, and then the fluid velocity increases as you go away from the surface. So if this thing is tilted, there's going to be a torque generated that will have this piece, high up piece, out of the flow. And this will point into the flow with the positively charged end. Think of this like a tail on a kite. And so, in this way, the flow can direct the direction of motion, and then the ultrasound and the chemistry can push either forward or backwards. And so, knowing that this is always going to go lighter end forward, we can make 2 different flavors of this. So, if we make rhodium gold, for example, rhodium is the lighter end. If the flow is going from left to right, this is now, the flow is turned on, but not the ultrasound. If we turn on the ultrasound, the ones that are scooting along the bottom have these false trails written in, and you see that they know which way the flow is going, and they go upstream. And then we can reverse that by having one, so in this case, the rhodium is the anode, and the gold is the cathode. If we do this with gold ruthenium, now the ruthenium is the anode and the gold, gold is the anode, ruthenium is the cathode. So flow and catalysis, again, oppose each other, but now the flow and the acoustics are driving the same way, and so these will go downstream even faster because the 2 forces are going together. So we can start to engineer this kind of stuff where these things can be trained to follow the flow. Finally, one of the disadvantages of these rod motors is they require an acoustic node that we made in this cell, with a standing wave, and that's hard to do over a long distance. If we really would like to use these, say, deep in the body, we shouldn't be using resonant ultrasound. We should be using traveling ultrasound waves. And the way to fix that is to make a different kind of motor, where we have a cavity, basically, that traps a bubble. So we make a hydrophobic inside, a hydrophobic outside, and trap a bubble here, and we rig it so the size of the bubble has a resonant frequency that's right around megahertz, right where medical ultrasound imaging works. And these work very nicely. So this is showing us propelling some of these things and making the pen motor by the little dark thing is the motor. It's pushing around tracer particles into the shape of the pen motor. We can direct these things where we want them, and these things are super, super fast, hundreds of body units per second. We can also have these are made by direct writing, 2D lithography direct writing. These are made by self-assembly and are much smaller and sort of do the same thing. And so we can make movies of these things. They do all kinds of complex behavior. There is a force, an image force, that makes the bubbles want to stick to whatever surface we have. We have to tilt them to get them to move. But we can, with collections of these, we can actually start to assemble them into what we call swimmer molecules. And you see a lot of linear ones and triangles. For the 6 ones, it's rare actually to see a hexagon like that. It's more likely to see a flat like that. Okay. And then finally, if you get more clever about how you design your swimmers, and this is something we're doing with Sherry Kagan's group at Penn, we can make these little, Sherry can make these lithographically, these little spider-like things that either they have chiral arms that either are in the same plane as the body, or they are up out of the plane of the body, like a chiral spider. And then if we suspend these at an acoustic node, what happens is originally they're all in every possible orientation, but fairly quickly they actually phase separate into groups of spinners that are all spinning either clockwise in one phase or counterclockwise in the other. And then they also go up and down depending on where the legs are. And so you can actually see this phase separation happening in real time all with autonomous swimmers that in principle don't know about each other except through interactions in the fluid. And this has been modeled by Sharon Glatzer's group at Wisconsin, where they've done all the fluid mechanics and shown that this is exactly what you expect to see things pushed together. They phase separate into different phases. So we're not sure what that's ever going to be good for, but that's kind of fundamental. Okay, and that's sort of where we are in this project. So what I've shown you is that we can propel micromotors and nanomotors various ways. That if you have interactions between them, either electrokinetic or acoustic interactions, they start to assemble in interesting ways. I didn't talk so much about pumps, but anytime we can make a motor, we can make a pump too by immobilizing the motor. And I just showed you a few examples, but now there are many other ways to help these things. And we're starting to see interesting applications, especially in diagnostics, I think, of these things. I think there are experiments going on with in vivo drug delivery, not in our lab, but in other labs, and this is coming on. Now, if we want to make real robots that can really develop swarm intelligence, we're going to need a few more things. We're going to need them to be able to sense their environment better, to communicate with each other, and have simple logic operations on the board. in addition to power. And that may sound like a lot, but individually, we're starting to see some of these things evolving. We're just really still. Okay, so it just remains for me to thank the generations of students who worked on this. I mentioned Wally, who got us started, and some of the early students are not on this, not represented here. This is Tiger Wei Wang, who did the first acoustic experiments with us and Mauricio, and he's now a professor at Harbin University, Shenzhen. The acoustic motor team is the students up here. I think I mentioned Nate and Vince, who are undergraduates. Suzanne, who's now a professor at UNC Charlotte, and Lee Chang is a grad student working on this. And then Jeff is a guy who's now worked on, taken over on the acoustic motors and designed spinners in collaboration with Sherry. Our friends who helped with fluid mechanics and math, Francois Nadal and Mallouk at Cambridge, and then finally Shannon Hosler and Sherry Kagan. Sherry engineering the spinners, and Sharon doing the theory on the spinners. All of this work was supported by the National Science Foundation, both at Penn State and currently. And that's the end. So I thank you again very much for your attention.
Yeah, I got you.
Create the swimmers, ultimate swimmers in such a way that deliver something to a.
Can you turn?
Sorry.
It is on.
Okay. Sorry, I couldn't.
So you either impregnate or coat these swimmers in such a way that deliver, let's say, a drug or something to a particular area. And how does that affect its ability to swim?
Yeah. So actually, Joe Wong at UC San Diego is doing many of those drug delivery experiments. So the easy target is delivering stuff across the stomach lining. He's done this with mice. So they can drive the pointy robot into the stomach lining and deliver drugs across that membrane. We've already demonstrated this. I mean, our dream would be to be able to inject some of these guys to power them where they need to go by ultrasound, to image them by ultrasound, and have them do their job. There already are some interesting results on antithrombosis, not from mechanically propelled or acoustically propelled, but from magnetically propelled motors that kind of showed, where they load the antithrombosis drug onto the particle. It goes through the bloodstream and finds the blood clot, and then they turn on the oscillation of this thing with magnetic field, and it actually does a better job of clearing the blood clot than if they just deliver medicine alone and don't turn it on. So these things are starting to happen. That's not our expertise, so we prefer to do more of the physics and then let the people who run mice colonies and things like that do the biological stuff.
Thank you.
There's another group in China that's done something really interesting, which is to basically wrap these things inside erythrocyte ghosts. So then the motor propels this thing that basically looks like a red blood cell. The immune system doesn't recognize it. But you can motorize the immune system thinks it's a red blood cell.
Well, I'm interested, there's some work with full nanoshells that they get to tumors, but it's kind of random how they get there. And then they since the body is relatively transparent as you're around, they can have those nanoshells absorb the energy that these nanometers are going to be revealed. Can you deliver your both nanorods?
Right. Yeah, that's right. So yeah, Naomi House at Rice has developed these gold nanoshells that will photothermally kill cancer cells. And Safed El-Sayed also did this with just the same kind of tiny, same shape, much smaller little rods that will enhance the local electric field and again, just get hot inside the cell. In principle, we could deliver them. I have to really think about the tiny ones that El-Sayed makes. These would be way outside the frequency range where medical ultrasound can propel. But it would be interesting to think about that. How you combine propulsion with photodynamic therapy.
So, in the systems like us, there are siderophoric molecules. And so I'm wondering, are there problems you've run into or opportunities you've speculated as a function of the various metals that siderophores, famous examples of siderophores you can isolate from ocean creatures can strip steel out of stainless steel, or iron atoms out of stainless steel. Would there be possibilities for problems or opportunities targeting siderophoric molecules in the body, or some body?
Targeting molecules.
Yeah.
I don't know how you do that. We can make these things move in concentration gradients, and there are plenty of examples in biology where multi-enzyme complexes assemble through spooky forces that we now believe are actually catalytic chemotaxis. But I don't know about chasing specific molecules. All of our structures are much, much bigger than molecules, so hundreds of nanometers. But it is interesting to think about maybe enzymatic chemotaxis in that context.
Oh, sorry.
Thank you. Thanks for the talk, I really enjoyed it. I'm just curious, you talked about the experiment to see how the rods behaved if they were eating the bacteria when they approached surfaces. And you found out that they were actually moving super fast, and that led to all these other experiments. But did you ever find out how they were behaving when they
So
approached surfaces?
you're talking about the DNA, RNA hybridization experiment?
No, you mentioned with bacteria how they sort of coast down to the surface and fly.
Oh, no, we never did find out. Right. Yeah, because what they did, what we saw by accident was much more interesting. We had one of the very contentious arguments in this field at the moment. I think everybody believes in the catalytic chemotaxis, for which there's a very reasonable physical explanation. But there's also the question of whether, for catalytic enzymes, whether their motion, their diffusive motion, speeds up when they're turning over their substrate. So one of the experiments we did while I was in France was a larger length scale to look at tracer particles in our catalytic rods, how they transfer momentum to each other. And with micron-sized things, you can definitely see this effect. With nanometer-sized things, this is now heavily debated. And it looks like there is a length scale below which just doesn't happen.
Any other questions?
Thank you. I think your talk is really shows how important interdisciplinary chemistry, biology, and physics. So as chemistry professor, you can see how the physics plays a role in the research. I just curious, could you elaborate more about you have undergrad student, a graduate student, what's the background, how to the work together?
That's a really good question. So you have to remember that undergraduates, unlike us, they're undifferentiated stem cells. They can do anything, and they're happy to learn. So we break down the problem. We have collaborators who are real experts in the math and fluid mechanics, and then we get together and we decide what experiments to do. And the undergraduates happily do their part. So this figuring out the mechanism of acoustic propulsion of the rods was quite complicated. They had to make lots and lots of samples to fit the theory, and they did that. There are some other interesting scaling issues that we've worked out theoretically that have yet to be tested experimentally. And undergraduates have been a very important part of that, both making the nanostructures and also writing the computer code to help us analyze the data. So there's really no problem engaging, there's been no problem engaging undergraduates. We also had a high school teacher who came from a local school under the RET program, made a good contribution to this point. And we have had high school students come in, really simple to make these little rod makers. And they can make them, and they can do the experiments, set up the ultrasound, watch them go, and track them, and stuff like that. So yeah, no experience necessary.
Thank you.
Sorry, one more question. Some of the rods that you were talking about, you were making yourself, were more pencil shaped and that sort of thing. Is there a way to design the rods to be more aerodynamic or more, to flow more freely through a liquid?
Yeah, so you're asking about the rocket-shaped ones. Yeah, so apparently this is enough to break the symmetry so they really run fast in ultrasound. But in the models just basically had cylinders with rounded ends. And just differences in density along the rod. So the density effect is a dominant effect. And so far the theory has not considered what happens with corrosion. Actually, Eric Mallouk did have one paper where he made a sort of a model that, it's kind of heart-shaped model with rounded curves so that you treat it mathematically. But it was the current model is a purely viscous model, Reynolds number 0. And Eric's model, which fits some of the data, was a finite Reynolds number model. We think is probably wrong. Because all of the non-zero Reynolds number models predicted motion about 10 times slower than what we see. And the purely viscous model hits it bang on the middle just. So we have more faith.
Any other questions? Seeing none, why don't we thank our speaker again?